world-renowned a world-renowned biologist by the name of E.O. Wilson. If you know him, he once said this quote. He said that if all mankind were to disappear from the world will regenerate into a rich state of equilibrium. However, if insects were to vanish, the environment would collapse into chaos. So this quote shows just how important insects are to our ecosystem. However, insects are only a small part of our equation, and they are a part of the wider group of animals or invertebrates known as arthropods. So hi everyone, my name is Li Si, and today I'll be presenting on my research titled Turning Over a New Leaf. Does forest restoration also restore soil and live litter arthropod diversity? Okay, so earlier I mentioned about arthropods. So what exactly are arthropods? So arthropods or Arthropoda is a taxonomic group of animals which consists of four smaller groups. So namely hexapods, which refers to your insects as well as springtails. There's also chelicerates, which refers to arachnids such as spiders, scorpions, and mites. There's also myropods, which are talking about millipedes and centipedes, and also crustaceans. So in this project, I'm focusing on the terrestrial ones, so mainly isopods. So in total, to date, there have been over 1.2 million different species of arthropods discovered so far. So this is an extremely large number, and if you put them into a pie chart, you would see that arthropods make up over 80% of all animal species. And if you compare them to all living species, so if you include plants, fungi, bacteria, so on and so forth, arthropods actually make up more than half of all known animal species. So why did I decide to choose this group of animals? Well, there are three main reasons, and I'll be covering them in the next few slides. Firstly, arthropods perform crucial ecosystem services. So they perform roles in our ecosystems such as decomposition, improving soil quality, pollination and seed dispersal, and they are also a crucial part of our food chain. Some of them are hunters, or some are being hunted by other animals. So in short, we can summarize their importance to the ecosystem in one sentence. Although these arthropods do not have backbones, they are one of the backbones of our ecosystem. Okay, moving on. The second reason is that disturbances in our forests are known to affect the functionings of these arthropods in the ecosystem. Over the past 150 years or so, rapid urbanization and deforestation has caused Singapore, my home country, to have only 0.28% of our original forest left. However, fortunately, recognizing the importance of greenery and our forests, the country has been putting in place restoration efforts over the past 2 decades. So it started way back in the early 2000s, and more recently, it includes measures such as the Forest Restoration Action Plan by the National Parks Board, as well as the 1 Million Trees movement. And so with this, we would like to study how our arthropods are responding to these restoration efforts. Okay, moving on to the last reason, there is a severe lack of such studies in Singapore. So this study is the first of its kind in Singapore and possibly the whole of Southeast Asia. That is because existing studies tend to focus more on charismatic species such as butterflies, dragonflies, and even ants. However, in this study, we include the less charismatic ones, such as isopods, millipedes, so on and so forth. Some of them we have never even heard of before, and some of them have never even been discovered by science before. And so with this, we hope that this study will serve as a backbone for future research in the field, as well as in the entire Southeast Asia region. Okay, so for the research question in itself, does forest restoration also restore soil and live litter arthropod diversity? Well, we have two main hypotheses for this. Firstly, we predicted that the diversity will be the greatest when the forest is less degraded. So if you look at this chart over here, we sample across 4 different forest types, and you can sort of arrange them in a forest gradient in which goes from least degraded to most degraded. And with this, we predicted that the less degraded the forest, the more diversity there are. And for our second hypothesis, we predict that forest restoration efforts is indeed helpful in restoring the diversity and community composition. So once you introduce restoration efforts to an unrestored forest, we predict that the restored forest will be more similar to the secondary forest. Okay, so with this, let us talk about how I conducted this research. So my methodology can be split into 4 main phases. The first phase is sampling. For the sampling phase, we went out into the forest and we collected our soil and leaf litter samples At the same time, we also measured environmental parameters such as soil temperature, soil humidity, and canopy openness. After that, we bring our samples back into our lab, where we conducted the second phase, extraction. So using the Winkler extractor and the Burley's Talgren funnel, we extracted these arthropods and collected them in a jar of ethanol, which we then brought them down into the lab to conduct our third phase, identification. Using a microscope, we identified each specimen to their order level. So order is a type of classification for our arthropods and for living things in general. So to do this, we make use of identification keys such as the one shown here, which is a step-by-step guide to help you identify each specimen. After that, we will curate the more interesting and good-looking species and create a reference collection for the arthropods we found here in Singapore. With the help of our research assistant, we also digitalized some of these reference collection and produced an online reference collection for the arthropods in Singapore. So I have included a QR code here. Later on, if anyone is interested, I can go back to this slide to share more about this. Okay. And so moving on to the last phase, which is analysis. There were two main parts to my analysis. The first is to calculate the biodiversity matrix for our samples. And also, we did a more community level analysis to help us better understand the distribution of our samples across the different forest types. Okay, so now on to the results. Here are some of the interesting observations that we had. In total, we had 120 different samples, which yielded over 20,000 specimens, spanning across 30 different orders. So these are some of the exciting specimens that we have, but bear in mind that this is only a tiny fraction of what is out there. And we suspect that many of these specimens have never even been described by science before. Okay, so on to the results proper. In the first hypothesis, we predicted that the less degraded the forest, the more diverse it will be. And this is largely consistent with what we observed. So for diversity, you can define diversity in terms of two aspects. The first aspect is richness, which is how many different types of organisms there are. And then the second aspect is abundance, which is how many organisms there are. So looking at the richness, we can see that across our 4 different forest types, there isn't much difference in the richness across the samples. However, if you look at the abundance, we observe something very interesting. So you see that the less degraded the forest, the more abundant the number of specimens we got. We attribute this to the number of Acari specimens. So Acari refers to this taxonomic group of mites and ticks. So in the less degraded forest, we can have twice the number of Acari specimens as compared to the most degraded unrestored forest. And this is a good sign because Acari are indicators of good ecological health because they play important roles in nutrient cycling, and they're also indicators of the presence of other species, such as mammals. Okay, looking at the community composition, in the second hypothesis, we predicted that restoration efforts will indeed be helpful, and that our restored forest samples will be similar to that of the secondary forest samples. We will look at our analysis in terms of our soil and leaf litter samples using our NMDS plots. So NMDS is a technique used to visualize the similarity of our samples. So for our soil samples, we noticed that there wasn't any significant distinction between the communities of all the 4 forest types. However, if you look at the leaf litter samples, we observe some very interesting things. So firstly, you can see that the less degraded the forest, which is primary forest, the more clustered the points are. So that means the samples are very similar to one another. However, the more degraded they are, the more varied they are, meaning they are more spread out. Using our statistical analysis such as PermaNova, we also discovered that the primary forest samples are significantly different from the other forest types, while the other forest types such as the secondary, restored, and unrestored samples are not really distinguishable from one another. And so with this, putting all our observations and results together, here are some of our key takeaways from this project. Firstly, we noticed that disturbances of tropical forest, which is the type of forest in Singapore, from their primary forest state, has long-term impacts on the leaf litter arthropod communities. So meaning, if you disturb a forest from the primary forest state, the secondary forest onwards will be more distinct and different than that of the primary forest. Yep, and then looking at the primary forests themselves, as I mentioned before, they are especially distinct from the other forest types. Thirdly, we also note that forest restoration efforts does seem to be facilitating the re-establishment of our arthropods communities. However, since the restored forest community isn't really distinguishable statistically from our secondary and unrestored samples, we suspect that restoration of our communities is happening at a slow rate. And lastly, for future efforts can look into restoring secondary forests as well to facilitate the succession of our arthropod communities into that of the significantly distinct primary forest. And so with this, I have come to the end of my presentation. Thank you.
All right, once again, another fascinating presentation. I'll hand it over to the audience members and ask if you have questions for Li Si.
Hi, Li Si. I want to ask, before I ask my question, say that your presentation is really good. And I want to ask, how do you select different, like the study sites in your research?
Okay, thank you for your question. So for this research project, as I mentioned earlier, there were 4 different forest types: primary, secondary, restored, and unrestored. So I'll briefly go to the next slide, which will show the definition of these forest types. So to briefly go through, primary forest refers to forests that have never been disturbed or modified before. So they are your untouched forests since the 1800s. And then for secondary forest, it refers to a forest that has been regrown on previously modified land over the past 100 years or so. And then for our unrestored forest, it is initially dominated by this group of ferns called Dicranopteris. However, they underwent restoration efforts in the early 2000s. And lastly, the unrestored forest is simply is more or less the same as the restored forest. However, they have never underwent any restoration efforts. So with this, we can sort of arrange them into a forest gradient of least degraded to most degraded over here. Yep, and in the next slide, so this is the map of Singapore. Singapore is a really small country, and our biggest forest patch is at our central catchment here. So if we zoom into the central catchment, my research project focuses on this area, and these are the different locations that I got my samples from, and these are the different forest types in each of the locations. Yeah, I hope that answers your question. Thank you.
Fascinating. I'm sure we'd all like to ask a lot more questions of our panelists.